So welcome to the first Bio 112 lab. And today you're going to learn some skills that we'll be using several times through the semester. And we're going to teach you what a spectrophotometer is and how to use it right and how to use it wrong. And we're going to develop what's called a standard curve. So the theme of today's lab is really all about color. And we've chosen to work with this really pretty blue-colored solution. What this is a chemical called copper sulfate. Copper sulfate is used to kill algae. Sometimes it's used in swimming pools or ponds to control their growth. But it's harmless to us here in the lab at the concentrations we're using it at. So we chose it because of its nice blue color. So what I would like to sort of show you today is that this instrument called the spectrophotometer is able to determine the intensity of the blue color that is present in a solution. And in that way, we can put a number on the concentration of that chemical. So just looking at these three bottles, is it obvious to you that the shade of the color, the blue color is really the same, but that each of the bottles has a different concentration and the intensity of the color changes. So we'll talk a little bit more about that in a minute. So probably you could tell that this first bottle has the lowest concentration of copper sulfate because it's the palest blue, and perhaps this bottle has an intermediate concentration and this one has the highest concentration. But it's kind of hard to tell the difference. And in science, we do like to put numbers on things, and that's what the instrument is going to do. It's going to give us a number for the intensity of the color. And we call that the absorbance of that substance. So let's talk just a little bit about color and refresh your memory, or maybe you haven't heard this before. Let's talk about why things appear to have a certain color to us. So if we were looking here at this table, we can all see that it's green, it's dark green, and the reason it looks green to us is that all the wavelengths of light in the visible spectrum, and you can review the spectrum of light, you may have learned that in physics class, but it's the whole rainbow of wavelengths. This green table will absorb all the wavelengths of light except for green, and those get reflected back to our eyes, and so the table appears green. So that seems kind of strange, but it's important to remember that. So now let's look at a clear solution. Light of all wavelengths is shining through this solution. And it's kind of the same principle, but because we can see through it, the copper sulfate in this bottle is absorbing all wavelengths of light except for the kind of blue-green ones, and those come through and we see those. That's why solutions appear to have a certain color. So what we're going to do is place a small amount of the blue solution in a cuvette. We're going to wipe it, make sure it's clean because we don't want to get crud down in the instrument. And we're going to place the cuvette in the path of a beam of light. We're going to close the lid. We can set the absorbance at whatever wavelength we choose. And for this first exercise, we're going to choose all the wavelengths. We're going to start at around 400 and take a measurement called the absorbance. And we're going to crank it up to 500, 450 or 500, take another measurement and keep going. So take a moment to review which wavelengths of light correspond to which colors. The shorter the wavelength, the more end of the blue spectrum it is. So around 400 is blue, and then we go through green and yellow and orange and red, and the higher wavelengths in the visible spectrum, up around 700, are the red range. I kind of remember that because blue is close to ultraviolet, and ultraviolet are very short wavelengths that are very damaging. So the shorter the wavelength, the more energy it has. So that's sort of how I remember that. And at the other end is infrared, which you can't see, and those are very long wavelengths. So it's really simple. One thing to remember, though, is before you ever take a measurement with a spectrophotometer, you always need to kind of zero it or calibrate it. It's called zeroing or blanking. Sort of like when you use a balance and you put the weigh boat on and hit blank or tare, it tells the instrument what zero is. So before you start, and every time you change a wavelength on the instrument called the spectrophotometer, you always want to put a tube of plain water or whatever makes sense for the experiment or the measurement you're taking in there, and then you can hit a button called blank, and that will tell the instrument that that's considered no absorbance. So it's important to have that concept. So 2 things to remember: always make sure your test tube is clean. Always remember it goes in the right compartment in the instrument so that the light beam can go through it properly. Always make sure the lid is closed, and make sure that you blank the instrument with usually plain water or a plain buffer that has no color before you start. And then, and again, every time you change the wavelength, you need to put the blank back in and hit it once more. So what we're going to do is give you all some, they're joining us remotely, we're going to give you some numbers which represent absorbance values that we will obtain by measuring the absorbance of that blue-colored solution through the whole spectrum of wavelengths. And think about beforehand which you think will give us the highest number. So the absorbance values are going to vary from 0 up to about 2. It kind of can max out. So the higher the number, the deeper the color. And from this first exercise we're doing, we're not so concerned with figuring out the concentration of that blue color, but we want to see how the absorbance change as we go through all the different wavelengths. So let's just think about that. If it looks blue to us, is it absorbing any blue light? If it did, it wouldn't look blue. So I would predict that we'll see very little absorbance at the blue wavelengths. But if we crank it up into the higher wavelengths, the orange and red, it should make sense to you that it looks blue to us because it's absorbing the complement of blue. And so we would probably expect there to be lots of absorbance at the higher wavelengths. And see if that makes sense to you, and see if the numbers work out that way. And we're going to ask you to make an Excel graph, and instructions will be found on your handout that shows the absorbance of that tube of blue color at all the different wavelengths. It's called an absorbance spectrum. And once we do that, we'll be able to see the wavelengths of light at which that blue color absorbs most. And we're going to use that to make what's called a standard curve, which is our next exercise. So the next thing that we're going to do is use the instrument to distinguish among various concentrations of this blue color. You might want to do that. You can use very low concentrations of copper sulfate to kill algae in like ponds or swimming pools, but you wouldn't want to use too much. So it might be important to test the concentration or know it pretty precisely. So what we are going to be doing in class, and what we've done for you all, is created a dilution series using a known concentration as a stock, and we chose this one, I think 0.25 molar copper sulfate. So we know that because we've used chemistry and weighed it out and made a solution that we know. In your handout on page 3, there's a chart showing the sort of the recipe that we used to create each of these tubes. The first tube only contains water. The second tube contains 1 milliliter of the copper sulfate and 4 milliliters of water. The third one contains 2 mils of copper sulfate and 2 of water, etc. So as you go up, the concentration gets higher. So what we're going to create is what we call a standard curve. Now, if we were really good at pipetting, really accurate and precise and consistent and weren't sloppy, we should create a chart that we can use to determine the concentration of an unknown. So we'll see how good Melanie and Janet were at creating these tubes that are going to serve for us to make a standard curve. So looking at these, if I mix these up, would I be able to put them back in order, going from the least concentrated to the most? They look kind of the same to me, even though I know there's different concentrations in each tube. And so I think we're going to have to use the instrument, which I hope is more sensitive than my eyes at discerning the different intensities of color. So hopefully you've been able to create your graph, which we call a standard curve, and you have a line going through that graph. You'll be able to see how good your standard curve is by determining the r-squared value. If that r-squared value, which Excel can put on your line, is near 1, it means those numbers, those points on the line fall very close to the line. So there's not much scatter. But if they vary a lot and the r-squared value is lower, like say around 0.5 or even lower, it probably means that you were kind of sloppy, or we were kind of sloppy in setting this up, and that standard curve won't be so useful because we can't trust the numbers. But usually we do a good job with that. And what I want you to think through now, in my hand I have a colored solution. It has copper sulfate in it, but I don't know how much is in there. Melanie and Janet made this and they didn't tell me what the concentration is, and they're challenging me to figure that out using the spectrophotometer. So I could do a couple things. I could hold it up to some of them that I know and try to eyeball it, but I'm just not comfortable with that. So what I'm going to do is put it in the instrument, and I'm going to determine the absorbance value, and then I'm going to use that chart, that standard curve, and be clever and see how closely I can estimate the concentration of this unknown. There's a few rules though. Let's say this tube, they gave me a tube that was darker blue, more concentrated, higher absorbance than any of the tubes I used to make my standard curve. I wouldn't be able to use it because the rule is you can't extend beyond the concentrations you use to create your standard curve. And the reason for that is, at one end of the spectrum, the instrument will peg, it'll max out. And it might give me a number of 2 and not go any higher. So the instrument only has a range of values that it can measure, and I may have gone beyond that. That also happens at the lower concentrations. If I have so little in there that even the instrument can't pick it up, then I'm not going to be able to tell either. So I have to ensure that the absorbance of my unknown falls within the range of my standard curve. So keep that in mind. So I want to give you another little challenge. Let's say Melanie and Janet gave me a concentrated solution that was way more concentrated than any of the solutions I used to make my standard curve. It was really dark blue, but they still wanted to know the concentration. See if you can think through and be clever and come up with a way that you could determine that. So that's a little challenge for you, and write that in your handout. What could you maybe do to work within the range that is allowed to figure out that concentration?
All right.
Okay, so now Janet and Melanie have given me another challenge. They have given me a solution that has such a low amount of copper sulfate in it that I can't use my standard curve. So I'm going to try to be clever and figure out a way that I can measure really, really dilute concentrations of copper sulfate. So I'm going to do a little demo here. And what we've done is set up a series of dilutions. Very, very dilute copper sulfate. You probably can't see any blue color at all in there, at least not very much. Right, so the first tube has just water. The second tube has water and 1 drop of copper sulfate. The third tube has water and 2 drops. It says 3, 4, 5, and 6, but you really can't see the blue color at all. But because of the chemistry of copper, when you add ammonia to it, and I've got some really smelly ammonia here, we're going to add 1 milliliter of ammonia to each of these tubes and let it mix up. We're going to see that the two complex with each other, the copper and the ammonia, and it's going to create a really pretty kind of cobalt blue color. And we're going to see that this is kind of a trick that you could create a test that's really, really sensitive for measuring copper concentrations in solutions. So let's see. We always do this as a demo because it's so smelly. So that's just water. This tube had 1 drop. See the blue? This tube had 2, 3 drops, 4 drops, 5 drops, and 6 drops. Oh, ammonia. So now, if we mix these up, hopefully you can see that we've created a sensitive test for really low concentrations of copper. When you compare the hue, I guess you would say, if you're an artist, the hue of the blue color, it's really different, isn't it? So what would we have to do to create another standard curve to measure really low concentrations of copper sulfate? Could you sort of imagine that you can use chemistry and sort of clever tricks to create a test that could very accurately measure low concentrations of copper sulfate by adding another chemical? So we would need to set up make another standard curve, and maybe we'd even want to adjust the wavelength to coincide more with the maximum absorbance of this more purplish blue color. So we hope that this exercise sort of demystifies when scientists and technologists make tests for things. Often they involve color, often it has to do with making visible that which is not visible, and using color is an important way to do that, and hopefully this simple chemistry experiment here and demonstration will shed some light on that for you.